I'm happy to present him, Professor Dror Sharon from Hadassah, from the Hebrew University of Jerusalem in Israel and Hadassah Ophthalmology Department. Professor Sharon is a professor at the Hebrew University. He did his PhD in the Hebrew University at genetics, and then two fellowships of postdoctoral research, one at the Weizmann Institute and the other at the Harvard Medical School, under the supervision of Professor Dror, who is a world expert in retinal genetics. Professor Sharon is using high-end tools for studying novel retinal disease genes. One example is the identification of the FAM1618 gene as the most common cause of inherited retinal degeneration in Israel. He has several exciting projects, and some of them are related to development and application of genes and mutation-based therapy for various inherited retinal diseases, and we are very excited to have you as our speaker today, Dror. Thank you for coming.
Thanks, Yossi, for the invitation. So my lab is actually located at the Hadassah Medical Center at Ein-Kerem, affiliated with the Hebrew University. And I will talk today about a little bit about the basic human genetics, and then I will move on to theoretical calculations of carrier frequency worldwide, how many individuals are carriers of mutations in genes that can cause retinal diseases. And I think you will be surprised by some of this data. And then we will move on for therapies. And unlike the talk from last week, which was excellent, I will talk about gene-specific therapies and even mutation-specific therapies. So I think we also have this slide which shows us this excellent and beautiful organ that we love to work on. And the eye is a very ancient organ that has been evolved in multiple ways. And I think it was really problematic for people to explain how such an organ has been evolved. And even Charles Darwin, in his fabulous book, The Origin of Species, he really struggled with explaining how the eye has been evolved. And let's see some of his sentences. Now, I'm not going to read whole sentences because in his book, a sentence can start at a specific page and end 2 or 3 pages afterwards. So he started, when dealing with the eye, to say that to suppose that the eye could have been formed by natural selection seems absurd in the highest degree. So it couldn't even fit his own theory. And actually that book, the purpose of that book was to prove that his theory was correct. But if we move a few pages downstream in his book, he would start explaining how that happened. And he would say that if we assume that there are many different events, gradual events, from a simple and imperfect eye to what he called a perfect eye, then maybe this will help us to accept the natural selection acting on the eye. And although he concluded that the eye is perfect, I think he didn't have all the information that we have today. And today we know that the eye is not perfect. And I will show a few examples on how evolution can even improve our vision. So we all know the structure of the eye. I'm not going to go through all the details. And I think that we will all agree that the retina is the most important tissue of the eye. So my talk today will be about the retina, which is a thin layer at the back of the eye. And one of the issues that people struggled with when studying the anatomy of the eye is why the structure of the retina is as is. Why is it in upside down orientation? If we look at the retina, we will see that it is composed of different layers, and the photoreceptors are here at the back of the retina and actually the back of the eye. So the light needs to travel throughout the whole eye and then throughout the whole retina until it gets to the photoreceptors. But that's not enough. If we look at the orientation of the photoreceptor cell itself, it is oriented in a way that the the outer segment right here at the right end, which absorbs the light, is facing backwards of the light. So this is one issue that, of course, raises questions. And one of the problems it creates is that the axons of the ganglion cells need to go back to the brain on the right side, then therefore they need to cross the photoreceptor layer, creating the blind spot. There are few people who are trying to support the idea of the upside down retina by saying that since too much light is getting to the eye, some light is being absorbed by cells in the retina, and therefore the photoreceptors are not overwhelmed with too much information. So photoreceptors are really beautiful cells. We can see the rods and the cones. We will concentrate on the outer segment of the rods and cones and this little part here, which is the cilia, in which the gene that I will focus my talk today is expressed in. So another problem in the structure and the anatomy of the retina is the ratio between rods and cones. Human beings today barely use our rods, and rods exhibit about 95% of all photoreceptors in the retina. The reason for that is that we evolved from night creatures that do use the rods, but we don't use them anymore almost at all. And moreover, most of the genes that cause inherited retinal diseases are expressed mainly in the rods. Some of them are not expressed at all in cones, and yet they cause total blindness. So evolution can even improve that by changing the ratio of rods and cones. But during primate evolution, a new area have been evolved in the eye, which is the fovea. The fovea is a tiny area at the center of our vision, which is responsible for most our daily tasks. And the fovea, the human fovea that is located at the center is important, but it can even improve further. And if we look at the retina of birds or of prey, or even the pigeon, so on top of the central fovea that we see here, there is another region which is called the red area. You can see here by the orange color. The red area is a peripheral macula, and actually the bird of prey can focus simultaneously on two different spots at the center and at the periphery. Now imagine that humans could do that. It would be really excellent for our vision. It would be interesting to get back to Earth 100 million years from now and see if evolution did improve our vision or not. Probably Earth will not be here. And some people say that evolution doesn't function anymore on human beings. So it is a big question mark if the retina will be improved along future evolution. So if we go into a photoreceptors and we see the disc, on this disc within the photoreceptors, within the outer segment, happens a biochemical pathway of vision, which is called the phototransduction. It's a basic pathway that is common for many pathways in our cells. What is unique are the proteins, the proteins that are expressed in this pathway. Many of them are expressed only in photoreceptors. So we have a G protein coupled receptor, rhodopsin, or the conopsins. We have the G protein, which is transducin, and we have a cyclic GMP cycle to keep the channels open in the dark and close once a photon hit the photoreceptor. Now imagine that every protein that is taking part in phototransduction is encoded by a gene, and therefore mutations in many genes can cause inherited retinal degenerations, and this is very different from many other diseases. So the major phenotype I will discuss today is retinitis pigmentosa, or RP in short, which is a disease that usually the onset of the disease is at the age of 15 or 20 years, and it is progressing along the years. It can progress for 30, 40 years until the patient is totally blind. And what is nice with retinal diseases is that we have very strong clinical tools to assess the progress of the disease. Imaging, for example, fundus photos in which we can tell what is the state of the disease, or even OCT, which is replacing histology. We don't need to do histology, we just image the eye and we know how many rows of cells are in the photoreceptor layers and so on. And also ERG. In the ERG, we can actually measures the function of rods and cones separately and know what is the status of each group of photoreceptors. So let's move on to genetics. And until 1989, there was no gene known to cause retinitis pigmentosa, but we already knew that there are different inheritance patterns, recessive, dominant, and X-linked, meaning that there is more than one gene that can cause a disease. And in a dominant inheritance pattern, what we know is that a patient is affected because of a mutation in one of the two copies of the allele, and therefore the inheritance is vertical along generations, while in recessive, we have unaffected parents, and about 25% of their offspring will inherit two copies of the mutation, and therefore will have the disease. So both examples can be found in almost every inherited retinal disease. So unlike most of the human diseases that are monogenic or multifactorial, so in monogenic, we can say that all patients worldwide who have cystic fibrosis, for example, have mutations in the CFTR genes, and we have thousands of examples of monogenic diseases in humans. Other diseases are multifactorial, meaning that there needs to be a combination of variants in many loci, in many different genes, and in combination with environmental factors, the multifactorial disease will take place, and AMD is an excellent example for a multifactorial disease. But in inherited retinal degeneration, the story is a bit different. So the first gene to be correlated to RP was rhodopsin. Rhodopsin was found to cause about 25% of dominant RP cases in the US. But later on, different mutations in the same gene were found to cause recessive, meaning that individuals who carry these mutations are unaffected, individuals who carry recessive mutations, but individuals who carry dominant mutations are affected with RP, and these mutations are within the same gene. And later on, mutations in many different genes, and this is only part of the story, were correlated with different inheritance pattern of RP. And we can see here that the gene can be grouped by the protein function. Many of them are involved in phototransduction, in vitamin A cycle, and ciliary generation, and other factors. But RP is not the only it's not the only retinal phenotype. There are many different phenotypes, and the picture is getting more and more complicated with specific genes that can cause multiple phenotypes and specific phenotypes that can be caused by many different genes. And when a patient comes to the clinic with dominant RP, usually we cannot tell what gene is causing the disease until we do the genetic analysis. So along the years, many different genes for inherited retinal degenerations have been identified. The current number is in the range of 250 to 300 genes. And as you can see, the number is going up almost every month. And one of the issues that prompts the genetics that accelerated genetic identifications of IRD genes was actually the Human Genome Project. And this is actually a pathetic announcement by Bill Clinton in 2000 when he announced that the human genome is completed, when only 1/3 of the genome was completed. But on top of that announcement, we can tell that there was an impressive accelerated number of sequences, and actually the scientific community doubled the number of sequences every 18 months. And parallel, there was a drop of the cost of sequencing that allowed many of us to use next generation sequencing like exome sequencing or genome sequencing. And this tool really allowed us to identify many novel genes for diseases, including IRDs. So let me now go into our data and show you how this revolution in genetics helped us to identify the causative genes in the Israeli and Palestinian populations. So my lab we set up the lab in 2002, and since then we are recruiting about 100 new families with inherited retinal degenerations. So the total number is over 2,000 families. And when we started recruiting, I expected that at a certain point, there will be a lower number of recruitment per year, but the number is still high and similar from year to year. What is unique to the Israeli population is the fact that no matter which retinal disease we are studying, autosomal recessive inheritance here in brown is the most common one. And the reason for that is, of course, consanguinity. When we ask the patients that are recruited in the study, are your parents related or not to each other, then 75% of Muslim patients were originated from consanguineous families, probably all Bedouin families, 35% of Arab Christian families, and in the Jewish community, it depends on the ethnicity, but in the Jewish community, these numbers are going down along the years, while in the other communities, they remain more or less at the same level. If we compare the data from Israel to other countries in which this data were published, we can see again that autosomal recessive inheritance here in green is much higher in Israel comparing to any other studied country. And the high rate of autosomal recessive diseases, what it causes is a high prevalence of a disease. And about 5 years ago, we published that the prevalence of non-syndromic retinitis pigmentosa in Israel, and actually in the vicinity of Jerusalem, is 3 times higher than what reported in Europe and the USA, and stands at about 1 individual out of 2,000 with non-syndromic RP. If we include all IRDs, then the prevalence is much higher at about 1 in 800 individuals. At the beginning of the project, we realized that we cannot work alone on these projects and we need to work in consortia. So we established the European Consortium for Retinal Diseases. This is more or less in 2005. We started as 5 genetic labs, and now we are about 25 genetic labs from Europe, Israel, Canada, and the US. We are meeting once or twice a year and sharing very sensitive information, very sensitive genetic information. And the reason for that is that we realized that we identified the vast majority of common genes, and we are now left with genes that might affect only few families worldwide, and therefore we need to join forces to be able to prove that a novel gene is actually causing the disease, and this collaboration resulted in very nice publications. Later on, and it was even more complicated, we established the Israeli Consortium for Inherited Retinal Diseases, in which not only genetic labs, but also ERG centers and a bioinformatic lab take part. And we published our major paper 3 years ago, and we were able to determine the distribution of different phenotypes in our population. So RP, as I said before, is the most common one, most common phenotype, almost half of the cases, but this is true for many other countries. What is unique to our country are the relatively high prevalence of achromatopsia and stationary night blindness, which are common in Israel due to founder mutations in specific populations. We also could determine the frequency of each of the genes. Some of the genes can be found in every population you will study. ABCA4, H2A, and EYS are very common in every population, but what is again unique to the Israeli population are the FAM161A gene, which is the most common cause of RP in Israel, and it is rarely found in RP patients worldwide. And when it is found in other countries, in many cases, these are Jewish patients. A very interesting story is CNGA3. CNGA3 is the most common cause of achromatopsia in the Israeli population, and not only in humans, but about 10 years ago, a sheep model was identified with a mutation in that gene, and it was treated with gene therapy, and even 7 years after treatment, it is pretty stable, and the phenotype looks pretty good. And I will also talk about RPE65. This is the only gene in any human disease for which gene therapy has been approved, and this is really a breakthrough in treating retinal diseases. So one of the first questions we wanted to ask is, first of all, when we analyze genetic data in publications, is the information accurate or not? And Mo Hanani, who was a PhD student in my lab, she's very talented in generating databases and analyzing them. And we wanted to answer this question. And the first thing that we saw is that a paper has been published in scientific reports claiming that a mutation in a gene called OR2W3 causes autosomal dominant RP. A very nice title for a paper. However, since I did my first postdoctoral fellowship on olfactory receptor genes, I knew that OR2W3 means olfactory receptor, and that 75% of our olfactory receptor genes are actually pseudogenes. They lost their function, and we actually are losing our sense of smell along evolution, maybe because we are gaining better visual function. But anyway, it was clear to me that when you are analyzing olfactory receptor genes, you are likely to find sequence changes that might be interpreted as pathogenic mutations, and that this has nothing to do with retinitis pigmentosa. So we wanted to ask the question, what percentage of genetic variants that are reported to be responsible for a disease are actually pathogenic? And the analysis was clear and surprising. I'm not going to go through this whole process, but just to show you the conclusions. So 10% of the reported pathogenic mutations are actually not causing a disease, they are mistaked. And about 20% of all genes, and this is even worse, 20% of the genes reported to cause dominant RP are not causative. So with this information, it's really difficult to believe what is published in the literature, and every geneticist needs to take care and analyze the data very cautiously. The next step was to try and estimate what percentage of people, of unaffected people, are carriers of a mutation that can cause inherited disease. And we started analyzing that when I was in a postdoctoral fellow at Harvard, together with Carlo Rivolta. And what we did, since we didn't have a lot of information in 2003, we estimated that the number of RP genes is 45, and that every gene contributes the same to the disease, which is, of course, now we know is not true, but using this totally theoretical mathematical approach, we were able to say that 1 out of 6 individuals is a carrier of a mutation that can cause RP, or mutations that can cause recessive RP. This is a very high value, which was not really accepted well in the community. And therefore, together with Mo Hanani, we try to analyze the data more accurately with the information that we have today. So we analyzed, first of all, the Israeli population. You can see here the different Israeli ethnic groups, Bedouin, Druze, and others. And what you see here on the y-axis is the frequency of unaffected individuals in the population who carry mutations that can cause RP, and the frequency is relatively high. Let's talk about two groups, the Ashkenazi population, for which we have the most comprehensive data. So 1/3 of the population carry a mutation, and the light part of the bar here represents the genes that were not identified yet. So it is likely to be higher than what we estimate today. Even more surprisingly are the Arab Muslim population in East Jerusalem. Based on the current genes, 47% of them are carriers of mutations that can cause a retinal disease. And once all genes are identified, we predict that most of them will be carriers. This is really an amazing number of individuals. We then moved one step forward and analyzed not only the Israeli population, but worldwide populations. And this is a very complicated analysis that has been performed by Mo, in which we analyzed over 250,000 of variants that were published in 187 IRD genes. And the first step was to determine which of these variants are indeed pathogenic. And we found that 10,000 of the variants can cause a disease. This study was published in PNAS last year. And then we tried to estimate, based on these variants, what percentage of the population are carriers and how many are expected to be affected by the disease. And as you can see here, in these purple bars for each of the populations that we studied, about 1/3 of the human population worldwide are expected to be carriers. It's an enormous number of 2.7 billion individuals that are carriers of recessive retinal mutations. And the expected number of affected individuals is over 5 million. But I think that this pie chart represents the picture in a better way. What we see here is the different populations based on the number of individuals of each population. So Asian population is actually dominating the globe, and most of the individuals originated from Asia. And if we look at the number of non-affected carriers, non-affected individuals who carry a retinal disease mutation, then the number is impressive. Over 500 million individuals only in East Asia are carriers, and over 1.5 million individuals are affected. This number are high in every scale, and probably carriers for IRD mutations are very common and more common than any other disease that has been reported so far. So let's move now to a different story. This is a story of the FAM161A gene from gene identification about 10 years ago by Dikla Bandach-Rosenfeld in my lab, to characterization and therapy of mutations in that gene by Avigail Beryoskin and Pradi. So as happens many times in genetics, different labs you sometimes have the same result. So when I joined a meeting in 2009 and met a colleague of mine, Carlo Rivolta, and we discussed our project, we realized that we identified mutations in the same gene as the cause of RP. Until that time, the gene was unknown, and nobody knew its function, and nobody knew if mutation in that gene can cause a disease. Now we know that this is the most common gene for RP in the Israeli population. So we identified using classical human genetic project. We identified 3 different mutations, one in an Arab Muslim family, which is a very rare mutation, but the 2 other ones are relatively common, and they are shared by the different ethnicities among the Jewish population. One of them is a nonsense mutation, and I will get back to it when I talk about the therapy of FAM161A, and the other one is a frame shift mutation. Both of them are founder mutations in the Jewish population. So at that time, we were pretty naive to think that the gene produces only 2 proteins. So when we analyzed RNA of the gene in the retina, we saw 2 different isoforms, one that contains all exons and one that skips exon 4. We were surprised to see when we have done evolutionary analysis that the exon that is skipped, exon number 4, has the highest conservation. What you see here is a plot of a sliding window. Each dot represents an average of 30 of the conservation of 30 amino acids along the protein. And we compare here the human protein to other species, and a summary of all the data. So you can see here that the exon that is skipped here and is expressed in only minority of the transcripts is actually the most conserved part of the exon, of the gene. And when we analyzed the protein isoforms, we could see different expression levels. So the major isoform, which is lacking exon 4, is expressed mainly in the rods, and the minor isoform expressed mainly in cones, but also in rods. It is expressed in the inner segment, not in the outer segment of the photoreceptors. Now we know that this protein is a ciliary protein interacting with 50 other ciliary proteins in the retina. Recently, we were able to characterize about 100 patients with FAM161A RP mutations. This is done mainly by Eyal Banin, who is a close collaborator of mine. And we could see that although the patients have classical RP features, some features are unique to FAM161A, including the lack of bone speckle pigmentations in the fundus that we don't see here in this patient. We do see in some patients, but this is a hallmark of RP in other genes. We then wanted to create a knockout mouse model, and this year we reported of the clinical characterization of a knockout. We knocked out the major exon number 3 of FAM161A, And we could see that, first of all, there is a progressive degeneration of photoreceptors and progressive loss of visual acuity. The mice are totally blind at the age of 8 months. And this is a relatively slow degeneration process comparing to other mouse models. Also, ERG, as you can see, is deteriorating until the 6-month point, in which we don't see any function of the retina. And by histology, you can see that the retina is losing the photoreceptor nuclei. At 6 months, we barely see any photoreceptors left. And not only the photoreceptors are affected, but also the inner retina, the number of nuclei are going down along the years. So it seems like 1 month, which look pretty nicely packed photoreceptors, 1 month would be a good target for gene therapy. But as happens many times in life, there are problems in gene therapy in 1 month, and I will describe 2 of the problems we are facing right now for gene therapy. One of them can be seen only by electron microscopy, and this study was done together with Uwe Wolfram from Mainz, Germany, who is an expert in electron microscopy for retinal structure. What we can see here is the wild type outer segment, wild type retina of a 1-month-old mouse, and you can see that the outer segments are nicely packed and nicely oriented in the retina, but in the mutant, already in 1-month-old mice, we can see that the outer segment are disorganized and lose their orientation. If we zoom in, we can see that, as we would expect, the cilia is affected. The connecting cilium is wider than the wild type, and the cilia is shorter. We quantified it and show that there is a significant difference in the length of the connecting cilium. And maybe a hope from this analysis was the fact that next to disoriented photoreceptors, we can see here the outer segment of a photoreceptor. Next to it, you can see an intact and healthy photoreceptor. And hopefully, these photoreceptors will be able to obtain the gene therapy and improve visual function of these mice and the patient. So one problem that we are facing is that although the retinal structure by histology looks intact and nice at 1 month old, when you check for details, you can see that the photoreceptors, many of them, are already damaged and might not be able to accept the gene therapy treatment. Another problem is that, as I said earlier, we were naive about our thinking of gene expression and splicing of the FAM161A. FAM161A is a very complex gene in terms of splicing. In the beginning, we published only the exon 4 story, which is an alternatively spliced exon. But when analyzing RNA-seq data from the retina, we could see that many transcripts go into introns, both intron 4 and intron 5, and some of them even skip within the intron. So in our retina, many transcripts are being produced but cannot be functioned because they do not contain all the domains of the FAM161A protein. This is a summary of the results. We can see here that about 13% of the transcripts are non-useful, and the major isoforms are presented here. And this is causing another issue for gene therapy. And gene therapy, as you know, is gaining a lot of focus lately because of the RPE65 and the Luxturna story. And actually today in Israel, every new child who is diagnosed with biallelic RPE65 mutation is eligible for gene therapy of Luxturna, which is, as you probably know, almost $1 million in cost. Gene therapy is now developed for many other genes, and these are only the genes that are clinical trials, and many of them are developed in mouse models. So gene therapy is really gaining a lot of attention, but it holds some problems. And just to highlight a few of them, when we're talking about genes that are alternatively spliced, like FAM161A, and it is known that more than 50% of the human genes undergo alternative splicing. So we have critical problems when we come to treat the retina. should we inject each transcript separately? And if we do so, what if both of them or all of them are vital for function, then we are losing a lot of effect. But if we mix them prior to injection, we don't know the real ratio for injection, and we don't know if they have a different, sometimes we don't know if they have a different expression pattern. So these are real limitations. One way to go about this question is to inject within, to insert into the AAV a mini gene construct in which we include the introns and let the cell decide which transcript to produce. But usually these introns will be too big for AAV, and modifying the introns is likely to affect splicing. So these are the issues we are dealing with right now in performing gene therapy for FAM161A, and therefore we and others are looking for other methods that will be applied for such diseases. So instead of treating the DNA, maybe treating the RNA might have a better effect. And this is a project that we are performing together with Shai Ben Arroya and Erez Levanon, who are here in the audience. And we are trying to recruit an enzyme called ADAR, adenosine deaminase acting on RNA, to modify a single nucleotide in the RNA transcript that will correct the mutation. So ADAR can modify adenosine to inosine, which is recognized as guanine, and maybe correct a mutation. And this is an example of untreated cell showing only the wild type nucleotide. And after treating with ADAR, we can see that another nucleotide appears because of the ADAR treatment. So basically, ADAR, what it does is to modify a single nucleotide in the RNA molecule. Where can we use it for treating diseases? So let's look at the protein, part of the protein, and we can see the codons here. So if there is a mutation modifying an amino acid to a stop codon, then we will have a truncated protein that is likely to be degraded in the cell. If we can recruit ADAR, and ADAR will modify one nucleotide, then we will have a different amino acid at the same spot, but we will have a full-length protein, and this might be correcting the phenotype and establishing a treatment. So what we generated so far, and this is an ongoing project, is a plasmid which includes two different color coding, the mCherry and GFP, and the gene with the mutation inserted in between. So if you are correcting the mutation, then you will have both colors expressed in the cell. Otherwise, you will have only mCherry, and this can be done pretty quickly in a 2-day experiment in which we can apply ADAR and look under the microscope to see if a correction has been performed and to what level. So this is an ongoing project which we are applying on various mutations that are either specific to the human Israeli population or some mutations that are common to the worldwide populations. Another way to treat retinal mutations are not at the RNA level, but at the protein level. So this is the translational read-through mechanism in which we are trying to correct nonsense mutations. So if there is a wild-type protein, we will obtain a full-length protein that is translated. But if there is a stop codon, a premature stop codon, we will again get a truncated protein, which is likely to be degraded by the cell. But some compounds, including ataloran, which is probably the most well-known compound in read-through therapy, can, instead of a stop codon, insert an amino acid that will create a full-length protein, again, with one difference, one missense change in that protein. How can we know that such protein is functional? Let's go back to FAM161A. So what I said earlier is that the FAM161A is expressed in the cilia. This is a cilia photoreceptor. And if we grow fibroblast cells and starve them for 2 days, the cells will grow a cilia, as you can see here. These are fibroblast cells from a healthy individual. But if we do the same and take a skin biopsy from a FAMA61A patient who is homozygous for a nonsense mutation, the cell will not grow a cilia following the starvation. And now we can apply a tolerant or other compounds to the medium and see if they grow a cilia. As you can see here, these are treated cells, and the level of correction, and it's not only that we created a full protein, but we created a full functional protein that can almost totally correct the phenotype. So we are, to summarize the therapy part, we are using 3 different levels of therapies: at the DNA level by AAV gene therapy, at the RNA level by other, and at the protein level by read-through therapy. So to summarize, there is currently no cure for the vast majority of inherited retinal degenerations. And the meaning is that millions of people worldwide are losing their vision gradually, and they know that they are going to be blind, but there is almost nothing that can be done about it. And gene therapy, therefore, holds great hope to treat these patients, to stop the degeneration process, and hopefully improve the vision by getting back the cells that are present in the retina but are not functional. And families in which we identify the gene can use the information and are using the information for genetic counseling and planning of the family. And we already saw in some of the genes, the most severe ones, that there is a reduction in disease prevalence along 20 or 30 years since the research has started. And if we look at the future, the genome of each one of us will be available, and we will be able not only to tell which mutations we are carrying, but which diseases we will develop later on in life, and hopefully to treat the disease even before the symptoms appear. So this gives a lot of hope, and I will thank my lab members for performing all the projects, and many of the collaborators, but mainly Eyal Banin, who is the PI. He is a clinician ophthalmologist, and he is diagnosing all the patients that are in this study. Denise Dalcarat from Paris, who is involved in the gene therapy, and also Ivan Arsenevich from Lausanne. Carlo Rivolta, who is involved in many of the projects. Shai and Erez, who I already mentioned. Uwe Wolfram, who is responsible for the electron microscopy study, and his wife, Christine, who is responsible for the read-through with the tolerance. And I would like to thank you all for listening.
Thank you very much, Dror. So we have some time for questions. Okay, so Dror, maybe you can tell us about the project that about the achromatopsia, something that is, what is new is this?
Yeah, so achromatopsia, first of all, the prevalence of achromatopsia in Israel is higher than anywhere else that has been reported in the world. And one of the genes, which is not the common gene worldwide, CNGA3, in Israel, 85% of achromatopsia cases have mutations in that gene. And a farmer in the north part of Israel realized that some of the sheep in his herd do not function at daylight. They don't go to have food. They don't follow their herd. And he contacted Professor Ron Ofri, who is a veterinarian ophthalmologist in Israel, and he diagnosed them with what he calls day blindness, which is another word for achromatopsia, and due to a CNGA3 mutation. And the first treated sheep with gene therapy, AAV-based gene therapy, happened about 7 years ago, and one injection in the eye was enough to restore vision of the sheep. And you can see very nice movies in which the sheep, which is blind, during the day, they hear the herd and they want to join them, but they cannot find the path because of achromatopsia. But after one injection, few months after injection, they walk just like a normal seeing sheep, and this is a long-lasting therapy for achromatopsia, and now it is being tested on human patients.
Is it already in human?
It is now in a clinical trial.
In Hadassah?
Hadassah takes part of it. It's a multi-center all over the world, but it is not that easy. I mean, it's again, gene therapy can be tough to achieve. Results are not very promising right now, but there are modifications that are being taken, and hopefully it will be more promising in the future.
Okay, thank you. Any more questions? Okay. Dror, I have another question. I'm not sure that if you are aware about it or anything, but do you have any updates on non-retinal gene therapy? I know it's not your direct area.
The only one that has been approved by FDA is Luxturna right now. Yes, there are clinical trials in other diseases, but I don't know what is the current status. But it is a promising field. Yes.
Yeah, it is. There are a few clinical trials. Okay, thank you. Okay, Dror. So thank you very much for this exciting talk. We are looking for a new result in the coming years to see what's going on in this field, which is booming, yes. Okay, thank you very much. Let's all thank again, Dror. And see you later. Sharon, do you have any updates on the next week's seminar?
Yeah, actually, I haven't checked, I have to be honest. We will meet again next week for another talk. I'll publish the details. Maybe it's Serge Picot, but I'm not sure about that. So I will, but we will meet at the same time, same day. We'll send information and updates.
Okay. Thank you very much, all. Bye-bye.
Thank you.
Thank you. Thank you very much.